is 60-65% proteins and 30 to 40% lipids. In the cell, in the eukaryotic cell, two different types of endoplasmic reticulum have been described. One of these is called rough endoplasmic reticulum, and you see the cisterns of this rough endoplasmic reticulum everywhere here close to the nucleus of the cell. And even you can see that the endoplasmic reticulum, the granular or rough endoplasmic reticulum, is in communication with the outer nuclear membrane. And the spaces inside the cisterns of the endoplasmic reticulum is in communication with the perinuclear space between the outer and inner nuclear membrane. It is called rough or granular endoplasmic reticulum because under electron microscope, small granules are visible on the surface of these flattened cisterns and sacs. These small granules are ribosomes, which are attached to the surface of the endoplasmic reticulum. So when on the surface of the endoplasmic reticulum ribosomes are attached, this endoplasmic reticulum is called rough endoplasmic reticulum. And here you see some cisterns which lack ribosomes on their surface, and this represents the smooth endoplasmic reticulum. And you can see that rough continues into the smooth endoplasmic reticulum. And all these cisterns are interconnected with each other and form a huge enclosed space in the cell for serving different functions. And there is a special functional division of endoplasmic reticulum called transitional endoplasmic reticulum. This is an area from which transport vesicles are budding, and these transport vesicles go to other parts of the cell, mostly to the Golgi apparatus, but some of them are going to other organelles or other areas of the cell. So these are the two major types of endoplasmic reticulum, rough and smooth endoplasmic reticulum. That's how these two types of endoplasmic reticulum look under electron microscope. Here you see an electron micrograph which shows part of a cell. That's the cell nucleus here on the upper left corner, and the rest of the cytoplasm is filled with these cisterns of rough endoplasmic reticulum. And it is rough endoplasmic reticulum because if I zoom here, you can see these small granules which are being attached to the surface of these flattened cisterns. And this is replicated, this is replicated here on this diagram. And you can see the cisterns of rough endoplasmic reticulum with many granules, ribosomes on the surface of the cisterns. And here in the middle, the smooth endoplasmic reticulum. This electron micrograph here represents the smooth endoplasmic reticulum, and these are vacuoles and cisterns lacking granules on their surface. Here you see a part of one mitochondrion on the electron micrograph. And once again, see this continuation of the cisterns of rough into the cisterns of smooth endoplasmic reticulum. So they represent one unite internal space in the cell. Which are the functions of the endoplasmic reticulum? Why endoplasmic reticulum is so important for the cell? Here again you see the cell nucleus, the rough endoplasmic reticulum, and smooth endoplasmic reticulum. First, endoplasmic reticulum is a region in which extensive processes of synthesis occur. Smooth endoplasmic reticulum is the site of lipid synthesis and carbohydrate metabolism. Phospholipids are produced in the smooth endoplasmic reticulum. Rough endoplasmic reticulum, because of the presence of these ribosomes on the surface of the rough endoplasmic reticulum, is related to protein synthesis. proteins are synthesized in ribosomes, but immediately after the translation process, these newly synthesized proteins goes inside the cisterns of the rough endoplasmic reticulum, where these proteins undergo the process of so-called post-translational modification. And then these proteins are directed either for secretion out of the cell, for incorporation into the plasma membrane, where they become membrane proteins, or they stay attached to the membranes of the endoplasmic reticulum, or stay as enzymes inside the cisterns of the endoplasmic reticulum, or become enzymes in another organelle that we'll discuss next week in our class next week, lysosomes. So these are the synthetic functions of rough and smooth endoplasmic reticulum. Second, transportation. Because these cisterns are all interconnected with each other, substances from one cistern can go into another, from rough into smooth endoplasmic reticulum, and in this way substances can move in the cell, but sequestered, separated from the cytosol. So they move inside the cisterns of the endoplasmic reticulum. Endoplasmic reticulum is also a storage area. More specifically, the storage function is typical for the smooth endoplasmic reticulum. And in smooth endoplasmic reticulum, in many cells, ions are being stored, sequestered from the cytoplasm. And last but not least, detoxification. This is again more attributed to the smooth endoplasmic reticulum, and that's where detoxification of drugs and alcohol happens. So, speaking about the functions of the endoplasmic reticulum, we see that endoplasmic reticulum serves quite various functions. But to explain these different functions, we must know the structure of the endoplasmic reticulum. And we'll see first the structure of the rough or granular endoplasmic reticulum. This is a bright field microscopic image from nerve tissue, namely spinal cord, medulla spinalis, and this is a special staining called the Nissl stain. Nissl is written with capital N because it is the name of a German pathologist, Franz Nissl. He introduced this staining method, and in this staining procedure, a basic dye is used. The dye is called Crusyl violet and has a violet color. And because it is a basic dye, it binds with the ribosomes on the surface of the rough endoplasmic reticulum, and we can see the regions in the cytoplasm where we have many cisterns of rough endoplasmic reticulum in the form of these purple patches or spots called Nissl granulations, like the spots of the tiger. This is another in the ventral horn of the spinal cord, medulla spinalis, and this neuron is famous, and all neurons are famous as having many cisterns of rough endoplasmic reticulum. That's why Nissl stain is widely used stain to study nerve cells and to demonstrate ribosomes and rough endoplasmic reticulum in neurons. And here is an electron micrograph of another cell which has abundant rough endoplasmic reticulum in its cytoplasm. These are the liver cells called hepatocytes. Here you see these parallel membranes of rough endoplasmic reticulum with many ribosomes attached to the outer surface, both outer surfaces of these cisterns. So, rough endoplasmic reticulum has ribosomes attached on its surface. And ribosomes attach to the surface of the rough endoplasmic reticulum when they produce proteins, when they conduct the process of translation. And here, namely, you see the process of translation. You know that messenger RNA is the one which contains the genetic information, the code for the newly synthesized protein. Messenger RNA is transcribed in the nucleus from DNA and then exits into the cytoplasm where protein synthesis happens. And then messenger RNA binds with the ribosome and protein synthesis begins. These proteins, which are designated to enter into the endoplasmic reticulum, contain a short amino acid sequence, the so-called signal sequence of 15 to 40 amino acids, which signal the cell that this protein must be sent to the endoplasmic reticulum. And then in the cytoplasm, there is a soluble signal recognition particle, that's a protein, which recognizes these several amino acids and binds to this signal sequence. And then on the surface of the endoplasmic reticulum, there is a signal recognition particle receptor. And the whole ribosome is sent to the surface of the endoplasmic reticulum, and with this interaction between ribosome, signal recognition particle receptor, and another larger receptor complex called translocator complex, the ribosome becomes fixed on the surface of the endoplasmic reticulum. And we see on electron micrographs these ribosomes which are producing proteins located on the surface of the endoplasmic reticulum. When this attachment happens, then the signal recognition particle is liberated and the newly synthesized protein is directed through the translocator complex inside the endoplasmic reticulum. Here, on the inner surface of the membrane of the rough endoplasmic reticulum, there is an enzyme called signal peptidase. This enzyme cuts and eliminates the signal sequence. It's not needed anymore. It has fulfilled its function and now it is eliminated from the newly synthesized protein. And the translation continues until the whole protein is being synthesized. So the example here is that the protein goes inside the cisterns of the endoplasmic reticulum. This protein might stay here as a functional enzyme in the endoplasmic reticulum, or it can be packed into transport vesicles and sent to Golgi complex for further modification, and then from Golgi complex to the plasma membrane, and these proteins will become integral membrane proteins, or Golgi complex will pack enzymes of lysosomes into primary lysosomes and will produce these organelles called primary lysosomes. But some of these proteins might stay in the membranes of the endoplasmic reticulum as integral proteins there in the membranes of the endoplasmic reticulum and serve as translocator signal peptidases and some other such membrane proteins located in the membranes of the endoplasmic reticulum. Okay, so let's see the fate of this protein which enters into the endoplasmic reticulum. When the protein enters inside the newly synthesized protein, inside the cisterns of the endoplasmic reticulum, it undergoes an excessive processes of transformation called post-translational modification of the protein. In the endoplasmic reticulum, this post-translational modification of the protein consists of glycosylation of the protein. This process of glycosylation consists of attachment of preformed oligosaccharide of 14 sugars, 9 mannose residues, 3 glucose residues and 2 acetyl glucose amines to the amino group of 1 amino acid in the polypeptide chain. This amino acid is called asparagine. In biochemistry, and according to the genetic code labeling, in the sequence of proteins, asparagine is labeled with letter N. That's why here it is written N-acetylglucosamine. N-linked precursor is being attached. This means attached to the amino acid asparagine in the protein, in the newly synthesized protein. Okay. The process of translation is usually quite efficient, but sometimes there are mistakes. Rarely, but there are mistakes, and wrong proteins can be synthesized. Endoplasmic reticulum has very efficient machinery to recognize such wrong or improper proteins and then rapidly directs these proteins outside the endoplasmic reticulum into the cytosol, where these proteins are being degraded by a special complex, protein complex in the cytosol called proteasome. Remember this process. I will describe proteasome in detail next week in our lecture regarding lysosomes as one of the mechanisms through which the cell degrades unnecessary and improper proteins. So, after this glycosylation of the proteins, these proteins must be sent to other regions of the cell, to other organelles. Mostly this is the Golgi apparatus. And this transportation from the endoplasmic reticulum to the Golgi apparatus happens via transport vesicles. We'll discuss transport vesicles next week, but briefly, there are 3 types of transport vesicles. One is coated on the outer surface with clathrin. The second type is coated with cautomer, protein type 1, and the third type is a vesicle coated on the surface with cautomer type 2 and type 1 cautomers. So vesicles which are coated with cautomer type 2 are responsible for the transportation from endoplasmic reticulum to the Golgi apparatus. This is called an anterograde transportation. In opposite, from Golgi apparatus to endoplasmic reticulum, the transportation happens via vesicles coated with cautomer type 1. You will say, why we need to transport something in opposite direction? Well, the cell is quite efficient in its sorting and transportation processes, but there are always some molecules which are wrongly packed and transported, and Golgi complex has the ability to recognize such molecules which belong to the endoplasmic reticulum and returns them to the endoplasmic reticulum. And the processes of transportation are always bidirectional. Okay, so that's how molecules are being transported to the endoplasmic reticulum and from the endoplasmic reticulum to the Golgi complex. And here you see a scanning electron micrograph showing cautomer type 2 vesicles budding from the cisterns of the endoplasmic reticulum, and here cautomer type 1 vesicles budding from the Golgi apparatus, so-called cisterns of the Golgi apparatus. And that was about the rough endoplasmic reticulum. And now let's briefly discuss the structure and functions of the smooth endoplasmic reticulum. That's again an electron micrograph showing part of the same liver cell, hepatocyte, but this area is enriched in cisterns of smooth endoplasmic reticulum. Again, no audio. Okay, so you can hear me, yeah? No problem with the sound. Okay, so smooth endoplasmic reticulum, you see these small tiny vacuoles or longer cisterns located everywhere in this region. These black spots here, these are depositions of glycogen, not ribosomes. Ribosomes are not so large, they are smaller granules. These black granules are depositions of glycogen in the hepatocyte. The smooth endoplasmic reticulum is structurally similar to rough endoplasmic reticulum, but it lacks the ribosome docking proteins, this signal recognition particle receptor and translocator complex, so ribosomes are not bound to the surface of the endoplasmic reticulum. And also, when there is no protein synthesis, rough endoplasmic reticulum changes into smooth endoplasmic reticulum because ribosomes are only attached to the endoplasmic reticulum when they produce proteins. After that, they detach from the surface of the endoplasmic reticulum. Because smooth endoplasmic reticulum lacks ribosomes, and ribosomes are rich of RNA, which is an acid, that's why ribosomes and rough endoplasmic reticulum is basophilic and attracts basic dyes because of the presence of RNA, ribosomal RNA. But here there are no ribosomes, and smooth endoplasmic reticulum is not basophilic, it is acidophilic. And areas of the cytoplasm which have abundant cisterns, large number of cisterns of smooth endoplasmic reticulum are not bluish purplish in color, not basophilic, but eosinophilic, so reddish pink in color. Smooth endoplasmic reticulum, as I said, serves for detoxification. In the membranes of the smooth endoplasmic reticulum, there is a protein from the family of cytochromes. Cytochrome P450. That's the most important cytochrome involved in detoxification of harmful substances. And smooth endoplasmic reticulum of liver cells is enriched with this cytochrome, and most of the detoxification processes happen there in hepatocytes of the liver. Also, smooth endoplasmic reticulum has the ability to concentrate ions. Mostly, this is valid for calcium ions. And please remember, it represents a depot for calcium ions in many cells. Calcium ions, as you will see later when we discuss muscle tissue, are very important for the muscle contraction. And muscle cells store a lot of calcium ions in their smooth endoplasmic reticulum. That's why muscle cells have very well developed and abundant smooth endoplasmic reticulum in their cytoplasms. The smooth endoplasmic reticulum there is so abundant that it receives a new name. It is the same smooth endoplasmic reticulum, but because of its abundance and main function to store calcium ions, it is called sarcoplasmic reticulum, the reticulum of the muscle cells. And another function of the smooth endoplasmic reticulum is lipid synthesis, namely phospholipid synthesis. And you can see here, this diagram shows the process of phospholipid synthesis in the membranes of the smooth endoplasmic reticulum. That's how new phospholipids are produced. And then vesicles from smooth endoplasmic reticulum bound by membrane containing these newly synthesized phospholipids are transported to the plasma membrane. and they fuse with the existing plasma membrane and deliver new phospholipids to the plasma membrane and also to the membranes of other organelles, membrane-bound organelles in the cell. And smooth endoplasmic reticulum is involved not only in phospholipid synthesis, but also in the synthesis of cholesterol and other steroids, derivatives of cholesterol, like sex hormones, for example, or vitamin D, which is also derivative of cholesterol. But this happens not in the membranes, but in the lumen, inside the smooth endoplasmic reticulum. And that's the endoplasmic reticulum, one of the easiest but very important organelle for the cell. Do you have any questions about the smooth endoplasmic reticulum? Okay, so let's continue with the second organelle for today, mitochondria. Mitochondria are organelles which were described, they were one of the first organelles to be observed and described in the cell because mitochondria can be quite variable in size. They might be quite small, 0.1 to 0.5 microns. That's around the resolving power of the most efficient bright field microscopes. But some mitochondria can reach a length of 2 microns, which is quite large for an organelle. And Carl Benda was one of the first scientists to observe mitochondria in 1898. And we mentioned first Carl Benda because he was the one to name these organelles and to call them mitochondria. He combined two Greek words, mythos, which means a thread, and chondros, which means a granule. Why? Because sometimes mitochondria appear as small granules. In other areas of the cell, mitochondria might be more elongated, like threads. So he called them mitochondria. They resemble either a granule or a thread. But observation of mitochondria were done earlier than Carl Benda. In 1850, Koeniger observed these organelles and described them. Fleming in 1882 and Altman in 1890. But Carl Benda named them. Usually cells have a lot of mitochondria, and probably you know why, because mitochondria, together with other functions they serve, are the major source of energy for the metabolism of the cell. Mitochondria are where ATP is being produced. And most of the cells have quite many mitochondria present in their cytoplasm. And here you see examples of cells. Fibroblasts are cells of the connective tissue, and they might have up to 100 mitochondria in their cytoplasm. Liver cells, hepatocytes, more than 800, 25% of the cell volume. And see the oocyte, 300,000 mitochondria present there in the cytoplasm of this large cell. Mitochondria were described under electron microscope in 1952 by George Palladay, and Shostran. And under electron microscope, that's a mitochondria here, surrounded by rough endoplasmic reticulum. Under electron microscope, mitochondrion is seen to have two membranes, an outer membrane, so an outer membrane, an inner membrane which forms infoldings called cristae. Between the two membranes, there is a space called intermembrane space, and in the center of the mitochondria there is a space which is filled with a special substance and other elements there called matrix. And these are the major structural elements of this organelle. We have an outer membrane, you can see it here. We have an inner membrane which forms these finger-like projections towards the inside of the mitochondria, irregular, some longer, some shorter, some thinner, some broader. The number of the cristae is variable. So outer membrane, inner membrane with cristae. Between the two membranes, that's the intermembrane space or the outer chamber. And then inside of the inner membrane, we have the matrix. And that's the structure of the mitochondria. Briefly describing a mitochondrion at the exam, if you draw it and if you explain these elements, this will be enough to pass the exam. But if you wish a higher grade, you must upgrade the understanding for mitochondria by giving some more information about each of these major structural elements. And now we'll discuss first the outer mitochondrial membrane. This membrane is usually thick 6 to 7 nanometers, and it contains many voltage-dependent ion channels which are called porins. You can see these porins here. They form large channels. The diameter is up to 3 nanometers, which are permeable for uncharged molecules with weight of up to 5,000 Daltons or 5 kilodaltons. The outer nuclear membrane is very similar in structure and function to the plasma membrane. And because of this, there is a theory about the appearance of mitochondria as organelles and why these organelles have double membrane layer around them. It is supposed that during the process of evolution, mitochondria have been other cells, prokaryotic cells, which have been taken by the eukaryotic cells via the process of endosymbiosis. And when the cells, the prokaryotic cell is being engulfed by the eukaryotic cell, it is being covered on the surface by the plasma membrane of this cell. And then in the inside, we have the prokaryotic cell. So mitochondria with their two membranes. Together with these porins, the outer membrane also possesses receptors for proteins and polypeptides, which are being transported into the intermembrane space. And it also contains several enzymes like phospholipase A, monoamine oxidase, and this one is important, acyl coenzyme A synthase. Because the outer membrane is similar to the plasma membrane and it is quite permeable membrane, as we see, the composition of the intermembrane space is quite similar to the composition of the cytoplasm. So the intermembrane space, which is the space between the outer and the inner membrane, has concentration of small molecules such as ions and sugars similar to that in the cytosol. Protein composition is different from that in the cytosol because of the presence of these special transporters, porins and receptors, which specifically transport some proteins and do not allow passage of other proteins through the membrane. So protein composition of the intermembrane space differs from that of the cytosol. And here in the intermembrane space, we have the presence of a special molecule called cytochrome C. Cytochrome C is a special molecule involved in the initiation of a process quite important for each cell called apoptosis. Apoptosis means a programmed cell death. And mitochondria are the organelles which actively participate in this process of programmed cell death. Indeed, mitochondria are the organelles which decide whether the cell to live or die. If the cell is designated to go towards cell death and to disappear, mitochondria are releasing this cytochrome C from the intermembrane space into the cytosol, and this cytochrome C triggers a series of enzymatic reactions in the cytoplasm, eventually leading to destruction of the cell. We'll have a special lecture dedicated to apoptosis, and then I will describe in detail the role of mitochondria in this process. For now, it is important just to memorize this important function, the role of mitochondria in the process of programmed cell death. And later, we'll see how exactly this happens. The inner mitochondrial membrane. The inner mitochondrial membrane is quite different from all membranes in the cell. Because of this supposed origin of the mitochondria to be a prokaryotic cell taken by the eukaryotic cell via the process of endosymbiosis, the inner mitochondrial membrane contains special phospholipids which are not present in other membranes of the cell. This phospholipid is called cardiolipin. It was called cardiolipin because it was first isolated from the heart of a cow. So cor is the Latin for heart. Cardiolipin is the lipid of the heart. And this cardiolipin is an unusual phospholipid. This phospholipid contains not 2 but 4 fatty acids in its molecule, and probably that's why this inner membrane appears to be very rigid and unpermeable membrane, mostly because of the presence of this cardiolipin. And additionally, the inner mitochondrial membrane forms these irregular invaginations called cristae towards the matrix of the mitochondria. So the inner membrane is irregular, while the outer membrane is a smooth membrane. And these cristae, they become more numerous to allow for more special proteins to be located in these membranes of the cristae. Why? Because these proteins belong to the so-called oxidative phosphorylation chain, and that's where ATP is being produced. The more ATP is being produced by mitochondria, the more longer and numerous cristae these mitochondria will have. So, you see here the cristae of the inner mitochondrial membrane, and on very high magnification images of electron micrographs, we can see that on the surface of the cristae there are these like mushroom structures present. In the past, because the structure of these mushroom structures was not known, they were just labeled F particles or F1 particles. But today we know that this is indeed one of the complexes of the oxidative phosphorylation chain, the last 5th complex, which is the large enzyme called adenosine triphosphate synthase. It is a multi-unit protein, and it is a very large protein complex in the membrane. That's why we see these F particles even at the level of electron microscope. So the major function of the mitochondria is oxidative phosphorylation. and it happens in the inner mitochondrial membrane. And oxidative phosphorylation chain consists of 5 protein complexes which are conducting electrons to each other, and while they conduct electrons, simultaneously they pump at some levels protons in the intermembrane space, achieving very high concentration of protons in the intermembrane space and building a very high electrochemical gradient between intermembrane space and matrix. And then the last complex, adenosine triphosphate synthase, allows for these protons to return to the matrix and capture the energy of this electrochemical gradient into the macroergic bond of ATP being produced by the fifth complex, the ATP synthase. Matrix. Matrix is the inner portion of the mitochondria inside of the inner mitochondrial membrane, and crystals are dipped in this matrix. Matrix contains about 2/3 of the total protein in the mitochondrium, so they have a lot of protein. Matrix has a lot of proteins in it. These proteins are mostly enzymes which act in two very important biochemical pathways. One is the citric acid cycle, or the cycle of Krebs, and the other is the fatty acid beta oxidation. These are the two most important biochemical reactions which happen, biochemical pathways which happen in the matrix. So please remember that. But matrix is unique because it also possesses ribosomes, mitochondrial ribosomes, and it has small circular molecules of DNA, mitochondrial DNA, circular like the DNA in prokaryotic cells, not separated with its own membrane. And mitochondria can produce their own proteins. The human mitochondrial DNA has 16,569 base pairs and encodes 37 genes. 13 of these genes are for proteins. There are 2 for ribosomal RNAs to make mitochondrial ribosomes, and there are 23 transport RNAs. They participate in the process of protein translation, protein synthesis in the ribosomes of mitochondria. So these 13 mitochondrial peptides are integrated into the inner mitochondrial membrane to form some of the subunits of these 5 complexes of the oxidative phosphorylation chain. And under electron microscope, we can see in mitochondrial matrix these black granules, which are matrix granules representing storage of calcium in mitochondria. So not only smooth endoplasmic reticulum can store calcium ions, the matrix of mitochondria also is a storage area for mitochondria. And because mitochondria are unique, here I have summarized the main functions of mitochondria. First one, energy conversion, the citric cycle, the beta oxidation of fatty acids, oxidative phosphorylation, and production of heat, storage of calcium ions, apoptosis, steroid synthesis, and production of reactive oxygen species. Mitochondria are unique organelles also because they have the ability to fuse with each other, so 2 separate mitochondria can fuse to form one large mitochondria. And also one mitochondrion can divide to produce 2 identical new mitochondria. So mitochondria are capable of fusion and fission. So when the cell needs more mitochondria, existing mitochondria can divide. When the cell lacks the need for mitochondria, mitochondria can fuse with each other to form large, relatively inactive mitochondria. So that's about mitochondria. We are going to finish our lecture class a little bit earlier today. Do you have any questions about mitochondria? Our class next week will be in this virtual classroom here. So please join this classroom and we will be discussing the structure of Golgi complex, lysosomes, and ribosomes. I see a question here. Are you expected to know the Krebs cycle? No, you will study Krebs cycle in detail in your biochemistry classes. Now it is all enough to know which biochemical pathway happens where in the organelle, but there is no need to know the biochemical reactions. That's topic of your biochemistry classes next semester. Okay, thank you for your attention, and we'll meet again here in this virtual classroom next Tuesday, 8 o'clock in the morning Bulgarian time.